All right, guys. Hello, what's up? Once again, guys. Here today, we're looking at transport across the cell surface membrane. In our last previous video, we're looking at the actual structure of the cell membrane, and we only mentioned that the cell membrane actually has got one main specific function, and that one main specific function, we said it is to transport, or rather to control exchange of substances across the cell membrane, meaning to say not all substances are going to pass through the cell membrane. It has to be selective, and other substances might not be allowed to go through the cell membrane. Okay, so before I start to explain these diagrams that you're seeing here, which is the different ways in which transport occurs across the cell membrane, we have to start by looking at some of the reasons why selective control of substances across the cell membrane is very much important with regards to cell structure as well as the cell organelles. So the first thing, remember, the transport of substances is two-sided. There are substances moving out of the cell into the external environment by processes of secretion, whereby substances are secreting substances like hormones out of the cell. Besides secretion, we also have excretion. Now, when it comes to excretion, we are talking about the cell removing waste products of the cell. For example, when the cell undergoes processes like apoptosis or cell destruction, it also needs to excrete those waste substances. There are also substances that are moving inside the cell. This is a process that is called endocytosis. So endocytosis is whereby the cell is taking in substances from outside, like nutrients and all that stuff. It's also a reason why it's selective. The other use of cell membranes across is the idea of maintaining ionic gradients. This is very much important. We have to maintain ionic gradients. This is specifically seen by nerve cells, especially when it comes to the idea of maintaining ionic gradients. But generally, our main purpose of this video is to explain the different ways in which transport occurs across the cell membrane in terms of diagrams. So now let's look into the structures. Okay, so now guys, looking at the structures, we have 4 diagrams here. So I'll just label them. Start with this one, which is diagram number 1. We'll also have diagram number 2. I'll also, okay, with order, let me say this one so that you understand it. This is diagram number 2, and this is diagram number 3, and another diagram, which is diagram number 4. These diagrams are representing the different purposes by which transport occurs. I'll start with diagram number 1 and diagram number 2. Diagram number 1 and diagram number 2, they involve a certain type of process, a transport process that is passive. And diagram number 3, as well as diagram number 4, they represent type of transport process that is active. In terms of these processes, diagram number 1 is called diffusion, whereas diagram number 2 is called osmosis. Okay, diagram number 3, we call this active transport. And then diagram number 4, this is bulk transport. These are the names. What do we mean when we say something is passive and something is active? Something that is passive simply means no energy required. So we are saying no energy required. And then active means energy required. That's the meaning. So we have passive and active. Difference is that one requires energy, the other one doesn't even require energy. Okay, so let's start with diffusion. Diffusion is a passive process by which we are talking about what we call a concentration gradient. Substances are moving from higher concentration to a lower concentration. So I'll use this space to explain the whole idea. So let's say this is the concentration gradient. At this place we have high concentration, and here we have low concentration. So by definition, we are simply saying diffusion is the movement of substances from region of high concentration to a region of low concentration. What possibly might be these substances? These substances can be gases, they can be nutrients, gases and nutrients most of the times as per biological example. But however, diffusion actually even occurs in some other areas, like let's say someone sprays a perfume in one area of the room. By the time, maybe after a few minutes, you find out that the whole room is actually smelling the same as a result of diffusion gradient. So for you to understand diffusion easily, I'm saying think of it like something is coming from a high landscape going lower. Of course, this thing will just easily roll to the lower levels. That's simply the imitation of when something is moving from high concentration to low concentration. Like for example, we say the gas is a nutrient. However, you find out that diffusion is further divided into two. Diffusion, there is general diffusion. If you want more about this, you go to my previous video on transport. But just to explain, there is what we call facilitated diffusion, which is a specific type of diffusion. Facilitated diffusion simply involves transport of molecules from high concentration to low concentration, but through types of proteins called carrier proteins or channel proteins. In this diagram, we are represented with carrier protein. There's a slight difference between carrier proteins and channel proteins. Why? Channel proteins are fixed. They are just fixed proteins that are responsible for acting as a channel, which they in my previous video. But looking at carrier proteins, these ones are mobile, they are moving, they're always characterized by a rapid change in directions. Okay, so in this diagram here, it's representing as a very good example of facilitated diffusion. Facilitated of diffusion of what? I'll give a very good example of glucose. This is glucose. Some of you might even ask why some molecules that they are being transported by facilitated, others by diffusion in general. So you find out that for molecules to be transported by facilitated diffusion or even by carrier proteins and channel proteins is because of two things. There's the aspect of size and there's the aspect of structure. So I want you to pay close attention here. When looking at the aspect of size, we are saying the size of the molecule determines the type of transport of diffusion. To be specific, if the molecule is very big, it cannot fit properly in between there. It requires a molecule that can accommodate it. Specifically, we are talking about less than 100 Daltons. Daltons is simply the unit of measurement used to measure molecular sizes. And then in terms of structure, using the principle that like dissolves like and unlike charges attract, only structures that are uncharged and they are also non-polar are allowed to pass through the cell membrane freely. So in terms of size and structure, that means the molecule, for it to pass freely through the cell membrane without the need of a carrier protein, it has to have less than 100 Daltons as well as its structure, it should be uncharged and non-polar. So let's give examples for each. Anything that is less than 100 Daltons, for example, we have gases like oxygen, nitrogen. These are very small in size. And then again, in terms of structure, you find out that uncharged molecules like water and non-polar molecules like steroid hormones, all these molecules, they can freely pass through the cell membrane without the need of any carrier or channel protein. Why? Simply because they are small in structure and they can fit in between the phospholipid bilayer, which is the idea there. Okay, so now let's explain the actual structure that is here. This is a basic example of transportation of glucose. So I'll label it and say this is a glucose molecule. This glucose molecule, for it to be able to be transported into and outside the cell, it has to pass through carrier proteins. These carrier proteins carry a specific name. They are called GLUT transporters. GLUT transporters. GLUT transporters, or GLUT proteins rather. GLUT simply means an abbreviation which represents glucose transporter proteins, or glucose transporter. Okay, so here's the thing. The mechanism in which these GLUT transporters work, they act, or rather exist, in what is called the ping-pong state. Let me say it again, ping-pong state. So the ping state is also known as the open state. The open state, where the carrier protein is facing the external environment, ready to receive. So I'll say the ping here, or I'll say also is also known as the open state, where it's opening up for the glucose molecule to come. Remember, we said for transport to occur in diffusion, molecules are moving from high concentration, maybe say from this diagram, outside is where they are highly concentrated, moving inside where there is low concentration. That's what creates what we call the diffusion gradient. So we have the open state, which is the ping state, and you see after it receives, you see this arrow moving here, a glucose molecule will attach to the receptor side. Once it attaches, which is the next step. So here it's moving, and then here there is attachment. Once attachment happens, it will switch and face the other side, which we now call the pong state. This pong state, or sometimes we can call it the closed state, the ping-pong state. Now in the pong state, it's now facing the external environment, where in the next step it is ready to release. Once it releases the environment, it also switches back. That means it can only switch sides when it is when there is attachment. Without attachment, it won't switch. Okay, however, for a further extension, we're not going to mention that more, but GLUT transporter proteins have got types 1, type 2, type 3, type 4, based on the location of the cells. But remember, all cells require glucose. Now, this is basically what you need to know about facilitated diffusion, not the general diffusion. This is diagram. And we said that general diffusion, which is passive, is whereby the molecules are moving freely without the need of these carrier proteins and channel proteins. This is what basically you need to know about diffusion. Meaning to say, when the concentration gradient is at balance, diffusion is less likely to occur. Diffusion can only occur when there is a diffusion gradient. This is passive process number 1. So I'll tick here and say diffusion is done.